Can this window be cancelled? This window can be cancelled. This is the question and answer. Okay.
Good morning.
That's better. I can see both of you now.
Yes. Okay. Good morning, sir. Good morning, everyone. Welcome.
Good afternoon here in Australia and also in Malaysia, I think, isn't it? There's someone from Malaysia come in.
Yeah, among the attendees, a lot from outside India as well. We have attendees almost over 400 attendees till now, and we'll be expecting more.
Wow, that's amazing.
Okay, thank you, sir. So I guess we can start. And good morning to everyone. Welcome to the webinar of Tropical Foods of Metabolic Syndrome. I myself, Dr. Baby, an assistant professor in Department of Food Technology. from from Mizoram University, and the person sitting next to me is Dr. Himjyoti Datta. He's also an assistant professor from the Department of Food Technology. So we both welcome you today on behalf of our department. And then we would also like to welcome all the participants from various organizations, from various research institutions, and I think many research scholars, many scientists, and many academicians are present here with us today. And we would also like to thank our respected Vice Chancellor who have organized and conceptualized this kind of program and this kind of platform for everyone to interact together. So we welcome you, sir, to our midst. And we would also, before we begin, I would like to request our participants to kindly leave any comments
I just
Yes, sir. Hello.
Maybe I just would like to say, I would like to say good morning to Brown.
Yeah,
Oh,
good morning.
Brown, I'm Professor Sambhasivarav.
Good to hear from you again. It's been a while.
Well, yes, I'm fine, sir. How are you?
Very well.
Yes, sir. I just a little bit late in joining. I will be listening to your lecture. Afterwards, I will speak again.
Thank you.
Thank you.
Okay, thank
you.
Go
ahead, baby. Go ahead, baby.
Thank you, sir, for your presence. As I've mentioned before we begin, I would like to request all the participants to kindly leave any questions if you have in the question and answer chat box, comment box, not in the chat box. And then according to the time availability, we will try to pick up a few questions at the end of the session if our speaker permits. So we will be doing that. And today we have, for our webinar, we have our speaker with us here, Professor Lindsay Brown, who is a well-known, renowned scientist in the field of biomedical sciences. He is from the University of South Queensland, and he has over an experience of more than 30 years in the field of basic pharmacology and nutritional physiology. Have done many work and research in the field of functional foods for treatment of various chronic inflammatory diseases, many publications in reputed scientific journals, and is still one of the leading scientists and academicians in the field of biosciences. We welcome you, sir, to our midst. So we welcome you once more from the academic fraternity of Mizoram University, and the session is yours, sir. Now. Thank you.
Thank you very much for the introduction, and thank you for the invitation from Professor Sambhasivarav to actually present this talk. It's a pleasure to do so. So I'll share the screen now. Okay, so I assume that's now clear for everyone. Okay, so what I'll talk about today is tropical foods for metabolic syndrome. And as you heard, we've worked in functional foods for some time now, and basically at the University of Southern Queensland in Australia. On the picture, there are some of the different fruits that we've worked on over the years. And some of those I'll be talking about today, but a lot there won't be, but there's a wide range there and some really fascinating work. The University of Southern Queensland has 3 campuses, and the one that we work at is in Ipswich. And on the left-hand side there, you can see Ipswich is about 30 or 40 kilometres, so 30 minutes drive west of Brisbane, the capital city of Queensland. And then if you go further, one of the other campuses is in the main campus is in Toowoomba, and we have another one also in Ipswich at Springfield. The pictures on the right-hand side are the overall view of the campus. This campus was originally set up as a lunatic asylum about over 100 years ago, 120, 130 years ago, and was changed into a university about 30 years ago, and we've taken over about 5 or 6 years ago. One of the really interesting parts of the university, as in many places, is the library, and that the bottom picture is actually the library of the university with this wonderful feature of plants and even trees within the library section. It certainly makes it very different from the usual libraries. So what I'll talk about today is basically tropical plants, but also some of those may grow in some subtropical areas. So the map at the top there, the dark blue is the tropical rainforest area, and so heavily forested area. And I think if you look closely, you'll find that Mizoram probably just gets in or maybe just out of the map, bit hard to tell, but maybe it's on the borderline there. And then if you look at the bottom slide showing the humid subtropical areas, and I think Mizoram certainly gets into that area. So somewhere on that border. Now you can see that Australia has some tropical areas in the north, and then along the east coast where we are is a lot of subtropical areas as well. So much of the work that we are doing, I hope, would also apply to Mizoram. These are pictures obviously taken from Google of a range of plants that are, or the fruit that are available in India. I recognize many of them, but I'm sure, or I hope that you people recognize all of them, since some of those don't actually grow in Australia. But we're not looking really at the whole of India. We're looking at Mizoram in particular. And you probably recognize the picture at the top, I hope, as your capital city of Aizawl. And the map on the left, India has a number of the hottest hotspots for biodiversity, the Western Ghats, for example, also the Himalayan areas, and also the eastern area of the country, so Assam and the other sister states. Now we can all go onto Google and see a range of fruits that are grown in there. And some of those, yes, I certainly recognize some of them, but many of them, I'll leave it up to you to work out what they are. But the big question is, can they be used as functional foods? So functional foods we'll define very quickly as foods that provide nutrition, but can also prevent or reverse disease states. And the disease state we're going to look at in particular is obesity, but probably also diabetes. India is well known as a country where the obesity rates are high and increasing. And some of the numbers there from the different states, if you go into the arrow, sort of about halfway down the right-hand side is Mizoram. And it's saying there from 2019 that about 21% of males and females are overweight or obese. Just obese, so 21% are obese. The figures down the bottom are the prevalence of diabetes and pre-diabetes. That's the figure for diabetes. Mizoram is about the seventh one in from the left-hand side, and that's at 5.8% of the population are diabetic. That's 5.8% of the whole population. The figures also show that in males aged 55 to 64, it's around 25% are diabetic. Now, diabetes is an incredibly costly disease in the long term, and so doing something about that is really important. So we've looked at functional foods and whether they are useful as potential therapeutic options for metabolic syndrome. So metabolic syndrome, including hypertension and fatty liver and diabetes and a chronic inflammatory syndrome. There was a review that we wrote in 2015, I think it was, or 2016, 2015. And you can see the other two authors there. So the second author, it was a PhD student, then has since graduated from Sikkim, from the state of Sikkim, and so at least part of the Northeast. And the third author, Sunil Panchal, is from Nagpur. And they've both been working on these sorts of things with me, and a lot of the studies come from those people. But the last sentence in that slide is the one that we think is really important. So the concept that these fruits and vegetables will act as functional foods to reduce obesity and therefore improve health remains intuitive and possible rather than proven. So it's a great idea, it's logical, and therefore it's possible, but the evidence to support this is remarkably weak. And we would like to think that it's not. We'd like to think there's lots of evidence, but the evidence is really quite weak. So what we've done is look primarily at rat models and a rat model of obesity. So what we do in that is the time scale below. So that is Sunil actually in those photos, Sunil Panchal as one of my co-workers. And we take rats for 16 weeks and feed them a high carbohydrate, high fat diet. So this is a diet-induced obesity. It's not a genetic obesity for one very simple reason. Humans are not genetically obese. Humans are obese because of the food they eat in most cases. There are some genetic abnormalities, but very, very few. So at 0 week, we give, or we measure the systolic blood pressure, we do the oral glucose tolerance, and AC is abdominal circumference. Very easy to measure, take measure, costs about, well, the one that we got cost about 50 rupees. And I think all universities can afford that. We then treat them for 16 weeks and we start the intervention at 8 weeks. We have a control diet too. We start the intervention at 8 weeks, and then at 16 weeks, we do the heart function, the liver function, and a lot of work on the organs and tissues. So we only start intervention at 8 weeks. So this is a reversal protocol, not a preventive protocol. And the very reason for that is quite simple. Most patients will be going to their doctor or be sent to their doctor by their family when they are already obese. So they are trying to reverse an existing state, not trying to prevent one. I don't think anyone goes to the doctor and says, I think I'm going to become obese and therefore need treatment. So this is really trying to model what happens in humans of trying to reverse the state of obesity. The first studies we did were on a range of purple fruits. So we started with the purple carrots, which is the original carrots. Many of you may have seen them. They certainly grow well in northern India along the Himalayas. They originally come from ancient Persia, and they have spread along the areas of India because many times there have been invasions coming from that area, and some of the good things they brought were the fruits and vegetables. Purple corn is an indigenous corn in South America, maize or corn in South America. The fruit at the top is a Queen Garnet plum. So it's a variety of plum that has been developed here. We'll talk more about that. And as you can see, it's purple the whole way through. Down the bottom is another plum, which is an indigenous plum in this country, which I'll also mention. But there are a whole range of berries that also contain the same compounds, and that's the anthocyanins. And those ones there are mulberries, but virtually every other berry contains the same group of compounds. So what we've done with the Queen Garnet plum first, so this is certainly a subtropical. It needs a certain amount of cold weather to fruit properly. And so it's subtropical, especially in hilly areas. So when we look at those down the bottom, so on the left-hand side of the top, the graph looks at the body weight on the left-hand side, left-hand axis, and the days, so the 16 weeks down the bottom. And looking at the gain in body weight, the black is the control ones, the blue, the controls fed with Queen Garnet plum from week 8 onwards. The green ones, the red ones, sorry, as the high carbohydrate, high fat. So this is a diet with lots of fructose, lots of beef tallow, 20% beef tallow in there, and lots of condensed milk. And they gain weight. As you can see that over the period of 16 weeks, they put on roughly 200 grams in weight. And most of that is fat. So there is some muscle increase as there is in control animals, but most of that is fat. Now, from 8 weeks onwards, we have given those animals, without changing the rest of their diet, given them the Queen Garnet Plum juice. And as you can see, the green versus the red, there is a very marked difference. The rate of gain of body weight from the green versus the red is very different. And that means that at the end, there's well over 100 gram body weight difference between those animals. Next to that slide is the picture of the structure. So these are anthocyanins, and the main one is cyanidin glucoside, which is the most common of the anthocyanins. Below that is a figure of the blood pressure, systolic blood pressure in those animals. So starting at about 115 to 120 millimeters of mercury, so very similar to a young adult human. And in the control animals, that goes up a little bit with aging, maybe to 125 or so. The red one are the high carbohydrate, high fat fed rats, so the obese rats, and their blood pressure goes up gradually to about 155, 160 sometimes millimeters of mercury. But look at the difference with the green one. So the green one, where the diet is the same, except for the addition of the Queen Garnet Plum juice. And after 8 weeks, blood pressure is now back to control. That's the sort of difference we're looking at for a functional food. So where in those pictures there, where we're seeing a decrease in the body weight, mainly in the fat, and also a decrease in blood pressure. So those plums were developed in our local area here by the state government, Department of Agriculture and Fisheries, and they crossed about 1,000 different, or made about 1,000 different crosses of the plum, and to try and increase the resistance to fungal infection. And then someone had the bright idea to actually try it, and they were really nice. And so they did another 7,000 crosses, which is remarkable perseverance, but did another 7,000 crosses and didn't find a better one. So this is the one they went with. This all happened about 25, 30 years ago now. And there are now something around, I think it's about 200,000 trees of these in Australia, in different parts of Australia, producing these plums. They're also available in New Zealand, in South Africa, in Spain, I think California as well now. So they are going around the world. The plum is actually owned by the Queensland government. And so they get royalties from every grower everywhere in the world for that. So if we look at the cardiovascular and the fat changes in here, so the cardiovascular changes, the blood pressure we showed before in the previous one, the graph at the left is the diastolic stiffness. So this is a measure of how well the heart can relax to fill with blood and how well it can contract to expel blood. In the fat rats, the red one again, there is an increased stiffness of the heart. It's an increased collagen, but an increased stiffness. It's harder for the heart to pump and to relax to fill with blood. The green one again has basically come back to normal. There is no difference between the Queen Garnet treated fat rats or the control animals. If we look down the bottom, we see the total fat mass. This is with DEXA, dual energy X-ray absorptiometry. So the same procedure that's used in humans to look for bone density, can also measure bone density in rats, but also fat mass and lean mass. And you can see that that's come down dramatically. It's not quite back to normal. It's certainly decreased though. And the other graph is the abdominal circumference. Rats don't have a waist, so we can't use the word waist circumference, but abdominal circumference is a really easy measure. And it gives us something really important to be able to talk with people. So not only talking with those that work in this area, but when we go out and talk to the general community, this is a measurement that everyone understands. And that's also important when we're talking as scientists in the community. If we look at the plasma concentration, so the Queen Garnet plums, the cholesterol, the triglycerides, the non-esterified fatty acids, as measurements of the lipid concentrations in the blood. And you can see for all of those, the red one is increased. So the fat rats are increased and the green ones are basically back to normal. NEFA didn't quite get back to normal, but it's certainly a long way down. The bottom slide is plasma activity of AST, I think it is. It's either AST or ALT. As a liver enzyme released into the plasma from damaged cells in the fat rats. Yes, there is damaged cell, there is release of this enzyme, so the activity goes up. And in the treated animals, again, without changing diet, we brought that back towards normal. So liver function, blood lipids, cardiovascular function coming down towards normal without changing diet, but by putting the Queen Garnet plum within the diet. And you'll say, okay, that's fine, but we don't treat rats, we treat humans. Well, what happens in humans? Human trials are much harder to do, they're much more expensive. We've done a small trial with 15, 16 patients in each group of hypertensive, mildly hypertensive, overweight patients, and gave them the Queen Garnet plum juice. So it's on the market in Australia. And the bottle is 250 mils. So they've got 250 milliliters to take on a daily basis. The red line is a cordial, a raspberry cordial, so no active ingredient. What we were allowed to tell the people was that we're testing between two different fruit products. Raspberry cordial is not really a fruit product, it just has the coloring, but we can see the difference there. So patients treated with the raspberry cordial, the red color, did not change their blood pressure. So either systolic or diastolic was unchanged over the 12 weeks of the trial. But look what happened to the group that were given the Queen Garnet plum. A consistent decrease in the blood pressure. So going down over the 12 weeks by about 11 or 12 millimeters of mercury in the systolic blood pressure and about 8 millimeters of mercury in the diastolic. And you may say, well, is that really worthwhile? That decrease in 12 weeks is as good or better than any of the standard antihypertensive drugs will achieve in that group. These are people with blood pressure that's borderline hypertensive. These are ones that are saying, can we prevent them from going on to treatment? Not saying they need treatment as an emergency, otherwise they're at risk of a stroke or a heart attack or kidney failure or whatever. These are patients that are borderline hypertensive. So this evidence showing very clearly that it is possible to decrease the blood pressure by a food, by a juice in this case, and that it's possible to reduce it almost back to normal in a relatively short period of time. Now, the other question, did they lose weight? No, they didn't. There was no change in fat deposition, no change in body weight. We told them not to change their normal diet. We told them not to increase exercise, do everything. Rats are much easier to work with than humans, but we assumed that they didn't. But in 12 weeks, there was no change. Would this take longer to work? Possibly. We don't have the data for that. We simply don't know. And that work was published, as you can see down the bottom, Journal of Functional Foods last year. Are there other fruits? Are there native fruits, indigenous fruits from Australia that may also work? Well, one of them I'm going to talk about, and that's the Davidsons Plum, which is the picture on the right there, unusual tree where the fruit grows on the trunk. but there's a whole range of other things, most of them from my home state. So most in the tropical areas of Queensland, many of which I don't actually recognize because I don't go wandering in tropical rainforests, but that's where most of them come from. So there's this range of foods, and there is some evidence that they are health enhancing, but this has not been a major topic of study. And I think that's something that needs to be remedied. So we decided to work on Davidsons Plum. Davidsons Plum at the top there. The size of those you can see by the ruler that's next to them, a couple of centimeters. I think it's up to about 8 centimeters. But the centimeter side there, the mass spec below, or the HPLC mass spec, showing which of the compounds are in there. So there is the cyanidin, but it's with a double sugar in there, as well as the glucoside, the sambobioside, which has a glucose and a xylose, I think it is, in there. There is also the peonidin in there, which is another glycoside, very closely related to cyanidin, and its sugars as well are in there. The main ones, though, are the cyanidin ones. So this is compounds closely related to the Queen Garnet plum. And if we look at the results from here, we can see the body weight gain percentage on the left-hand side, markedly increased in the fat rats and has been brought down with the 8 weeks of treatment. The blood pressure has come down towards normal, and the diastolic stiffness has also come down to normal. Now, this was part of a PhD study by Oliver John, who is from Sabah, in Eastern Malaysia. And he is now back in Eastern Malaysia, but the work published last year. He also looked at the area of the adipocytes. So took the fat pads, did the histology of those fat pads, and looked at the size of the different cells within there. And so on the left-hand side at the top, the size of the adipocytes, they markedly increase. They've double the size in the fat rats, and that comes back down almost to normal. The distribution of size is much greater. The next one, so the red one showing the distribution in the obese rats, the fat rats, markedly different sizes, fewer at the low size, but many more at the bigger sizes. The green one has improved that, certainly not back to normal, but it's only 8 weeks. And then the bottom one, okay, I can't remember what the bottom one is, but it's also one, I think it might be the adipocytes, and the top one is the fat pads. If we look at the histology of these, so if we look at the histology of the heart in the first 2 rows and the fat pads in the bottom row, so in the heart, the first column are the controls, the second column are control plus Davidsons Plum, the third column are the fat rats, and the fourth one, the fat rats treated with Davidsons Plum. So if we go across, the big difference in the fat rats, so number C in the heart, are lots of inflammatory cells. So infiltration of inflammatory cells, the arrows pointing to them. And when you look at the fat rats treated with Davidsons Plum, there are virtually no inflammatory cells. There's still 1 or 2, but virtually no inflammatory cells in the heart. That's an anti-inflammatory response. Yes, we can measure all sorts of mediators, but the picture tells the story. In the second row, stained for collagen. So using a Picrocereus red stain. So the red color, the light red color, red background as well, but the light red color is collagen. See the marked increase around the blood cells, so perivascular collagen, but also in the tissue itself, in the fat rats. In the treatment, it's gone back to normal, or relatively back to normal. Down the bottom row, the adipocytes. So the size in the first two, the control treated, look at the difference with the K. So these are the fat rats, markedly bigger cells. It's all at the same magnification. And the treated animals have come back. They're not back to normal, but they are very markedly different from the fat rats only. So this is what a native, what an indigenous fruit can do, as well as the normal, subtropical fruits. This is a tropical one. If we look at the liver in this one in particular, so the top row is just the H&E staining, hematoxylin and eosin staining. And what we can see there, and again, the third column are the fat rats, lots of white spots. These white spots, if we look into the next row, these white spots are lipid droplets within the cells. In the second row, we've stained them with oil red O, and that's a selective stain for lipids. And you can see the big difference there compared with the others. The bottom one is a collagen stain, again, around the veins in particular, a large amount of collagen in the liver. But look at the fourth column there and compare that back to the third column. So treatment with Davidsons Plum, markedly reduced lipid deposition. So very few lipid droplets in the H&E or in the oil red O staining, the second row. And the collagen is also markedly decreased. Again, not quite back to normal, but markedly improved. Remember, they're still getting the terrible diet. But this is one thing we found on this one that was a pleasant surprise. We'd shown in the last couple of years that these fat rats develop all the signs of an osteoarthritis. So an obesity-induced osteoarthritis, which also occurs in human, very well known in human. And so we've done the histology of the knee joint, and that's in the top part of the picture. A being control, B being control plus Davidsons Plum, C being the fat rats, and D being the fat rats with Garnet, with Davidsons Plum, sorry. So we've stained it there to show the different parts of it. And I think you can see very clearly that there is enormous damage to the top layer in the Davidsons Plum, in the fat rat, so in C. Davidsons Plum treatment has brought that back towards normal. Now, that sort of change should lead to a very marked improvement in the symptoms of osteoarthritis, the pain. We haven't done that in humans. That's something that we are trying to get the funding to do, but that's something that would be really important. Do we have a functional food that, based on this sort of evidence, can be taken into treatment of patients and relieve the osteoarthritis, which certainly damages quality of life? The Mankin score on the bottom is a score of the damage of the tissue, and you can see the marked improvement in the green versus the red. Now, I realize that Mizoram is not doesn't have a coastline, but you have countries very close to you that do have, and tropical seaweeds are a major source of food in parts of the world. They can also be a real problem in parts. There are also freshwater macroalgae, so seaweeds are macroalgae. There are freshwater macroalgae, and they can be really important, in particular in cleaning up water from heavy metals, for example. They can also be eaten. The three different types, so there's a red, a brown, and a green algae, macroalgae. They can sometimes be a pest. That picture down the bottom is on tropical coasts in South America. There was a big growth of sargassum a couple of years ago, 4 or 5 years ago now, and it ended up being up to 1 meter deep on thousands of kilometers of coastline. And apparently it was markedly, when it decomposed, it was revolting. So it can be a real pest, but can also be really useful. And the only slide that I'll show on this, Aneema is a seaweed that is grown in the, or that grows in the tropical seas off North Australia. So the coral seed also grows in the Atlantic. And so we tested that, published earlier this year, as you can see. We tested that on our fat rats. And you can see in the body weight graph, a very marked decrease in body weight. When we look at the blood pressure, back down towards normal. When we look at the fat mass, also markedly decreased. So these are compounds that seaweeds that contain carrageenan, carrageenan being a fiber. So the most likely reason for this is a prebiotic effect of the fiber in the seaweed. There is a very small amount of omega-3 fatty acids, too low to actually do anything. So seaweeds as well can be a remarkably useful functional food. One that is something you may not expect. So looking at a waste product. So spent coffee grounds, and this is something we've also recently published, but spent coffee grounds. So what's left over after coffee is made. The coffee belt on the left-hand side there, sorry, the coffee belt of the areas that produce coffee, whether it's Robusta or Arabica or a bit of both, as in India. As you can see, Australia produces virtually no coffee. There is a little bit grown here, but it's something like 10% of demand. But the countries that produce the coffee are all in the tropical belt. And the major ones are countries like Brazil and Vietnam and Guatemala, Honduras, many of the countries. India also produces some really nice coffee in Tamil Nadu. But you can see where it comes from. But waste product from coffee is an amazing amount. So not only in the production of coffee, so the coffee pulp, for example, but also in the spent coffee ground. So what's left after the coffee, the beverage is made. And these are figures from Sydney. Sydney, as the largest city in Australia, has about 5 million as a population. And in 2016, it produced about 3,000 tons of waste coffee grounds. So this is the waste of the coffee. We're not talking about the cups, the plastic cups and all of those problems, but just the waste coffee itself. Of that 3,000 tons, 7% was used in worm farms and gardens. Great. But 93% ended up in landfill. So it's literally waste that is helping to damage the environment. So can we use it? Well, first of all, what's in spent coffee? Well, spent coffee contains melanoidins. Melanoidin is simply a Latin term for black things. It sounds much more impressive talking about melanoidins than talking about black things. And we don't even know the structure of most of them. There's probably hundreds of them, slightly different. We know that there's a lot of different compounds in there, and you can see the list down there, different sugars, and then unknown linkages, and then the brown one down the bottom that says unknown material. So there's still a lot that we simply don't know even what they are, but they're likely to be prebiotics. So compounds that are not broken down in the stomach, but are broken down in the large intestine and are used there as for metabolism by bacteria. So we did some work on coffee, the waste coffee, and this was recently in the newspapers here. And the top one were pictures that were taken, obviously, of me, and also some spent coffee from my kitchen, actually. that were taken and used on the Australian Broadcasting Commission story, so the national broadcaster, ABC. But our local newspaper, the Courier Mail, also printed the story. And just to mention there, on the left-hand side down the bottom, study reveals the perks of coffee. They didn't actually mention anything anywhere in there that it's the spent coffee that we're talking about, not the coffee itself. But they got most of it right. But the whole newspaper article is on the right-hand side. And as you can see, there's this lovely young lady as something like 2/3 of the space there. And if you look in the text, it says that this young lady doesn't actually drink coffee, but she uses it as a skin rub. And what's that got to do with the story? I have no idea. But that's obviously the way the newspapers will try and attract readers. But just going back to that, but I think it is absolutely critically important that we as scientists, that we as people working in nutrition, working on natural products, actually go to the newspapers, tell them what we're doing, and try and get it published there as well. We will get an enormous number of comments back from the community. And the one that I like the most are people coming up to us and saying, oh, we're so glad the university is doing work like this. We are part of the community. We are a public university, so we are paid by the government, the federal government in our case, central government for you, paid by them. And so we have a responsibility to explain to the community what we do, why we do it, and how it could be important for them. But let's look at the data. So the spent coffee grounds, this was published in FASEB Journal a couple of months ago now. Spent coffee grounds, again, the green versus the red. Look at the difference there. It's basically stopped an increase in body weight in those animals. And when we've looked at the lean mass versus fat mass, look at the difference there. So in the control animals, in all the animals, very little change in lean mass. So virtually no change in the skeletal muscle, but look at the change in fat mass. So this is then evidence that this is actually working as a functional food. We've done the histology as well, and very similar, column C or C, G, and K being the fat rats, and the fourth column being the fat rats plus spent coffee. And you can see the difference. Virtually no inflammatory cells, virtually no collagen deposition in the heart, and in the liver, very much decreased lipid droplets in the liver, as we would hope, and that's what's happened. We then got colleagues of ours at the University of New South Wales to look at the gut microbiome. And this is something that is quite different. And if we look at that in particular, what we see in the top right-hand corner, the ratio of firmicutes to bacteroides. And that has gone up in the, has gone down dramatically when spent coffee is being given, whether that's in the normal rats or in the fat rats. It's still gone down. And so that is part of the reason for the changes. When we've looked at the different bacteria in there, so in particular, there's 4 groups there. There's 6 rats in each group. So control plus spent coffee, fat rats plus spent coffee. And the big difference is in the fat rats, so HS with spent coffee. And we can see there the Clostridia, so the light green color, the Clostridia as a major one of the firmicutes has gone down dramatically in the fat rats. And Lactospiraceae has gone up dramatically in most, not all, 5 out of 6 of these rats. When they have put these into the statistical analysis, you can see that there's very marked changes occurring in the from the fat rats to the fat rats plus spent coffee, H to HS. And these sorts of changes are logical because what we've got in there are prebiotics. So prebiotics, again, being used as food for bacteria in the gut and therefore changing the composition of the gut. That's the active ingredient. So we've looked at a number of other tropical fruits, especially from Garcinia. And so the obvious question is that relevant to your part of the world? Whether you're in West Bengal or Bangladesh or Mizoram, or even in Myanmar or down in Malaysia. These are areas, the bottom graph on the left, these are areas that where Garcinia, where Malabar tamarind, Garcinia Cambogia will grow. Also the Western Ghats and Sri Lanka. But also the mangosteen, so Garcinia mangostana, growing in those areas as well. The top figure on the left is showing that these are tropical fruits. They will grow around the world, including in my state, in Queensland, in tropical parts of it. The next question is, do they actually do something? Well, let's go into look at the first one. This is a Bolivian mangosteen. It's called a Chacha in Australia. In Spanish, it's a Chachairu. And we published this 2018. This is a small fruit. And you think, well, why on earth are we working on Bolivian fruits? Well, there are 2 commercial orchards of these fruits in the world. And one of those is in North Queensland. By far the biggest one is in North Queensland. The other one is in Guatemala. And when we've looked at those, again, the HPLC mass spec, what we see is that the procyanidins are there, both B2 and A2. You can see the peaks on the right-hand side at the top, and that gives the color of the rind. So these are what's in the rind at the top, and the pulp as well. So what we've looked at basically is that we've tested both of those, and what we've seen is that the pulp does virtually nothing. It tastes wonderful, by the way. It's a lovely lemon lime sort of flavor. It's very refreshing. It's really great. The rind is waste. And like many tropical fruits, this is about 30% of the weight of the fruit. And so this is a lot of waste that's going around. And so we've looked therefore at, in particular, at the rind and seeing whether that works. And yes, it does. So the rind is the one with R. So the control, a Chacha rind, CAR. The control, a Chacha pulp in the middle. And the high-fat rats with pulp and rind below. So if we look at the bottom 2 there, for example, there's lots more collagen in the fat rats compared with the control, so C versus H. In the pulp treated ones, there's no difference from the fat rats, but there's a marked reduction in the rind treated animals. So again, a waste product that's actually useful. When we looked at the fat deposition in the liver, the control ones, there's a little bit of the pulp had a little bit extra fat in there, don't know why, but in the fat rats, it was markedly increased. Look at the difference with the fat rats with the pulp and the rind, so HAP versus HAR, markedly reduced. It's still there, there's still some fat there, it's not 0 or 100% in either of them, but it is markedly reduced. Another one of these mangosteens is the yellow mangosteen, normally grown in the tropical areas, so in particular in Indonesia and the Philippines, but also in North Queensland and even in Brisbane. It grows in, even though we are classically subtropical area, it grows well here. The weight we see a very different effect here. So the green versus the red again. These animals actually lost weight, and they lost weight because it suppressed appetite. So even in the control animals, so the blue versus the black in figure A. So we see that the Garcinia dulcis, and this was the rind in particular. The seed is very large in these, but this is the Garcinia dulcis, that the fat rats went up as expected. The green ones not only stopped growing, they actually lost weight, and then it stabilized. So this wasn't a toxic effect. We looked at the gut in particular, and there was no damage to the gastrointestinal tract. The adipocyte area, the ones with the fat rats had many more, a much wider range, including some very large cells. This markedly reduced in the treated animals, so the green versus the red there. Virtually none of the large cells and more of the smaller adipocytes. We've got some histology, and on the top row this time is the adipocytes. You can see the third one being the fat rats, markedly greater in size, and that is reduced with the Garcinia dulcis, the yellow mangosteen. The next one is the heart, and the inflammatory cells are markedly reduced again, so G versus H. And then the collagen with a different color system. The collagen again markedly increased in K, the fat rats markedly reduced in L. So these sorts of changes, the changes in the structure, the changes in the function, due to addition of a waste product. The liver there, again, what you would expect. The liver, the fat rats, so the third column, C, G, and K. So lots more fat droplets. Down the bottom, the oil red O staining that, and then the collagen in the middle. Look at the difference in the fourth column. Fewer of the fat droplets, as shown by oil red O in L. Also much less collagen deposition, so in the liver. So we've improved the structure of the liver. And we've also changed the gut bacteria again. So this is looking at the firmicutes and bacteroides in particular. So the 4 groups down the bottom, each 5 or 6 rats, depending on the group. So the control plus Garcinia dulcis, the fat rats plus Garcinia dulcis. So the fat rats versus the control, markedly different. So the firmicutes have increased, the green ones have increased dramatically. And that's been brought back virtually to normal by the treatment. The diet hasn't changed. It's simply in addition to the diet. The other graph there on the left-hand side is the ratio of firmicutes to bacteroides. And you can see in the figures there, so means and deviation, compared with the individual rats on the other graph there. So again, marked differences from a waste product from fruit. So the same picture, we're almost at the end, the same picture from earlier on. The fruits from the northeast of India. So which one of those is going to be the next superfruit? Which one of those are some of the people who are listening today? Which one of those fruits are those people going to develop and show that it has a functional food characteristics and that it can reverse cardiovascular or liver or the abdominal fat, for example? Will they also reduce the damage in bones, so the obesity-induced osteoarthritis? I don't know. That's why we do the science. And that's what I would suggest, is that the people listening in, or certainly the people from the Northeast states, you come from one of the great biodiversity hotspots in the world. I'm sure there's lots more things out there that I don't know about, but I hope you do. And testing of those could really show some quite dramatic results. And the last slide. Well, that's fine knowing this. How are we going to interpret, how are we going to put this into the diet? And are there other possibilities? Well, of course there are. And these are some pictures that have been taken, maybe borrowed, taken from Google. So the juice of the corkum, so Garcinia indica, on the top left-hand side. In the middle is a purple carrot soup with black cardamom in it. We studied the black cardamom, got the best quality black cardamom from Sikkim, and it showed that it was very useful. Green cardamom wasn't. Green cardamom actually was damaging, but black cardamom was superb. Different seaweeds, and the bottom, the green banana flour. That's something that is, it's not a waste product, obviously, but producing a flour from unripe bananas, less sugars, less simple sugars, more complex sugars in there, and it certainly works. The fruit down the bottom, I'll put the note on there because otherwise I'll forget the name. So Sapota or Chiku. Does it work? I don't know. Different fruits, kiwi fruit in there, but also different berries, including strawberries and mulberries, different melons in there as well. And last but not least, coffee. So not only the coffee itself, and there's really good evidence, there's hundreds of trials showing benefits of the coffee as a beverage, but now we have evidence showing that coffee as a waste product, so the spent coffee, can be a functional food to improve the obesity, to improve the liver function, to improve the diabetes in metabolic syndrome. Does it work in humans? We don't know yet. There's enough evidence, there's enough anecdotal evidence after I was on the ABC, quite a number of emails saying that, yes, we've tried it and yes, it works. And the taste of it is actually relatively mild. It will be a coffee taste, but if you are drinking the coffee to start with, this will be much milder than that. And so this is feasible. So what I would suggest is that in the area of Mizoram, but also in other areas around the northeast of India, but also in areas like Malaysia, there are an amazing number of indigenous fruits. There are an amazing number of fruits that are grown in the area that could be developed as functional foods. So not simply by anecdote, but by using well-accepted scientific methods to show that they will improve health. Thank you very much for your attention.
Thank you, sir, for the wonderful talk. So our attendees have put multiple questions, out of which, due to the time constraints, we shall be selecting only a few.
Okay.
So the first question from one of our attendees, what approach?
You want me to open up the Q&A?
Pardon?
You want me to open the Q&A?
Not required, sir. We are putting the questions from our side.
Okay, thank you.
We have selected out.
Actually, Brownji, actually there are questions and Q&A. Either you can read or our people can read and tell you, whichever is comfortable for you.
I'm happy for Dr. Dutta to read them because he will pick which ones are, which questions are the most critical ones. And I'm glad.
Then he will do that. He will do that. Yes.
Responsibility is fine by me.
Yes.
So the first question from one of our attendees is, what approach do you suggest for Indian food habits? India's diet systems are versatile and varies from region to region. How to choose out of them?
it's impossible to choose for one thing for every region, because they will be different. But one that grows very well in your area is Corcum, Garcinia indica. There are some products available mainly from the Western Ghats that are on the market for that and can be used, as the picture shows here, it can be used as a drink, for example. It's not possible to do include every one of those in the diet, but it's certainly possible to include some of them. And things like the green banana flour, for example, if that's replaced up to about 15 to 20% in making roti, for example, no one will notice.
Oh.
It won't change the taste, but it will certainly change the dietary absorption of the carbohydrates.
The next question is, can you suggest an ideal model diet to prevent metabolic syndromes?
The ideal model diet to decrease metabolic syndrome is to decrease calorie intake. So a reasonable protein intake, a decreased carbohydrate or the simple carbohydrates, so the fructose, the glucose, the sucrose decreased, and also less oil. Indian cooking can be incredibly oily. So decreased use of oil. So to actually take everything in moderation and combine that with exercise as well.
Can citrus or lemon really prevent weight gain or induce weight loss? The consumption of citrus.
Of citrus, yes, it can. We did some studies a couple of years ago with naringin, which is part of the peel of grapefruit, for example, but also is the bitter taste in grapefruit. So well-known citrus. And yes, that certainly worked. The question is whether whether a large enough dose can actually be given on a daily basis, or is this simply one of many compounds that are in a normal diet that can be used? So instead of saying you must live on grapefruit, for example, breakfast, lunch, dinner, and have one before you go to bed as well, but saying that this is one of the many parts of a diet that can be used to help reduce the obesity.
One last question, sir. What is your opinion on the existing controversy relating to coffee consumption and heart health? Some reports say that coffeestol in coffee beans increase LDL oxidation, thereby affecting heart health. So what is your take on that?
There have been a number of overviews of studies, and one of them had, I think it overviewed something like 170 or something trials. And that came out very clearly that an intake of 3 to 4 cups of coffee per day for an average person improved cardiovascular health. It also decreased the incidence of a range of cancers, and it also improved liver function. So on the average, that 3 to 4 cups a day. Yeah, I'm a pharmacologist by training. Anything can be taken in overdose, including coffee. But that limit of 3 to 4 coffees per day will certainly improve health in the long term.
Thank you, sir. So our Honorable Vice Chancellor, sir, is with us. If he wants to say a few words for our attendees, so we shall be glad. Yes, sir.
Actually, Brown, sir, I have one question. Now people are saying in COVID condition, many people are saying if you can take lemon, if you can take ginger, if you can take all these spices in a huge quantity, that will be giving some kind of relief from the COVID-19. So, there are several, as you mentioned, it is a big biodiversity hotspot. So, what are your suggestions for remedial measures of COVID-19 in this aspect?
That we have to start with science, not believing in anecdote.
Yes,
There is remarkably little evidence for any of those.
yes,
The evidence is very clear that people, that younger people and that people who are healthy
yes,
do not suffer as much from COVID-19.
yes,
And so that maintenance of good health is the primary intervention. And
yes.
that can, certainly fruits and vegetables will help that. Whether they stop the infection by itself, we don't know. And I would find that hard to believe. But if a person is healthy, they are much less at risk from that disease state.
Actually, I was listening in many reports. They say that if we can take this kind of vitamin A, vitamin D source of food materials, it will be good for control of COVID-19 very soon in a couple of days, like 14 days, 15 days. That is one. Your experience, I would like to know, because no, you are the authority in that area.
I would like to see a randomized clinical trial with that,
Yes, sir.
so that it's not just anecdote.
Yes,
Something like greater than 90% of people recover.
yes, sir.
Yeah, there's an old saying in English that when a person recovers, God causes the recovery and the physician takes the fee.
Yes, sir. It is.
And that might be what's happening.
Yes, sir.
Most people will recover with or without intervention.
Yes, sir.
It's the group that are going into intensive care. That's the group that we
And
need to.
the mechanism is not being explained by many people. This is what I was really wondering. They are saying that if you take this one, it will be getting relief. What is the mechanism involved in that? What is the mechanism been presumed, also not been known?
That's right. It's simply that, yeah, to believe
Yes,
that they're antiviral and
yes,
that they will act in the very short time frame for some
yes,
people to go from the initial infection to being in an intensive care unit
yes, sir.
is science fiction.
Yes, sir. It is really good. Actually, because your various examples which were actually in the biodiversity hotspots of this region, and how they are actually been very rich source of relief for various diseases, even to COVID-19 also. And particularly, I was telling our people, there are several tribal ethnomedicines here in this area. If it has been tried with hospital patients of this particular tribal ethno experience, it will be more useful for them, because you can avoid English medicines, and there is no English medicine also. So if the tribal ethnomedicines are being tried with the people who are asymptomatic people also, it can be easily, some kind of experiments can be studied. This is what my idea is.
Yes, that's very correct. And they've been tried for many years now. Many of those will be completely placebo effects.
Yes, sir.
And so doing proper trials will sort out which of those are placebo and which are likely to be effective. But
Yes, sir.
also, that's one source of new medications. Another
Yes, sir.
source of new medications
Yes, sir.
are the waste products, are
Yes, sir.
the things that we
Yes,
are
sir.
throwing away, and
Yes, sir.
as if they had no value. So the
Yes.
tropical fruits with their large
Yes.
rind, the large peel, the
Yes,
coffee
sir.
waste, which everyone will say, oh yeah, we'll just try to put it in landfill.
Yes, sir.
That's not a solution to it. But so there is that source of traditional medicine, it could be really effective, but
Yes,
there's
sir.
also this source of waste products, and
Yes, sir.
developing those in collaboration with the engineers, for example, with the pharmacologists, I hope, even with
Yes.
pharmacy students, will actually, because they're free to start with,
Yes, sir.
actually be a lot less costly than medications made overseas, wherever.
Yes, very good, sir. So thank you very much, sir. We are really, because many participants were appreciating the lecture which was given today. It is very useful and informative. More than 500 people, and some people were denied the entry into lecture hall because our limit is only 505. So many people could not join. Maybe sometime else we can spare us some time, and one more lecture you can plan. Maybe when you are free, you can just put me an email. So now I'm available for one more lecture. So then I can arrange one more lecture. Those who missed it today. Yes, sir.
Also,
Thank you very much, sir. Thank you very much, and very nice to see you again after almost 5 years. I think almost.
I think it's even maybe a bit more than that.
More than that, yes, sir.
You can ask Dr. Gupta to send me the questions as well.
Yes, sir.
And I will try and answer at least some of them and send them back as an email to him.
Yes, sir. Very nice, sir. Thank you very much, sir. Thank you. Bye-bye.
My pleasure. Thank you very much.
Thank you very much. Bye-bye.
Just one information for our participants. In case you require or anything, there is a YouTube channel, the Mizoram University channel. So there the session has been uploaded. And if you want to comment anything, give a feedback or anything, you can always go back to our session. And we once again thank you for all for participating, and thank you, sir, to our speaker. Clearly presented and very interesting session. We thank you, sir.
If you can send me some of those questions as well.
We shall be sending on the questions, sir.
And I'll try and do something on those over the next week or so. And
yes, maybe the questions you can copy and you can send to Professor. That is better.
Okay, okay, sir. Yes, sir. Thank you.
Thank you very much.